top of the hour. Let's get started. Welcome everyone to this Society for Immune Polymorphism summer webinar. I'm Steve Mack, moderating. I'm the Society President, and I'm very excited to introduce today's speaker, Professor Louise Boyle of the Department of Pathology at the University of Cambridge. Dr. Boyle's laboratory researches peptide antigen processing presentation by MHC molecules, and her group identified the TAPBPR protein component of the class 1 antigen processing pathway. Today, she will be presenting her work describing how TAPBPR and MHC class 1 polymorphism shape peptide editing. Before we start, we have a chat. If you have questions, please put your questions in the chat, and we'll be happy to address them at the end of the webinar. So please welcome Professor Boyle.
Thank you, and thank you very much for the invitation to give a webinar as part of the series. Yeah, so today I'm going to talk about TAPBPR and these MHC class 1 polymorphisms and how these two combined help to shape the peptide editing process. So MHC class 1 molecules are critical for immunosurveillance. So their function is to present peptides, a fraction of the cell's proteome, to T cells. And they're also recognized by natural killer cells. And as a result, these class 1 molecules, which are the most polymorphic molecules in the human body, are of major clinical importance, and particularly when we think of things like infection control and also in the recognition of tumours. And the MHC class 1 antigen processing and presentation pathway has been extensively studied over the past 3 decades. And we now have a very good understanding of the major players involved in this pathway, which help to assist MHC class 1 molecules to bind to peptides. So these peptides are typically generated in the cytoplasm via the proteasome, and in a fraction of the peptides that are generated can be imported into the ER lumen via the TAP transporter. The class 1 molecules are known to fold with the help of chaperones such as calnexin, calreticulum, and ERP57. And then once the MHC class 1 molecule has bound to beta-2 microglobulin, it's then in a peptide receptive conformation and can be incorporated into the peptide loading complex. And this peptide loading complex consists of the TAP transporters pumping the peptides, the MHC class 1, calnexin, calreticulum and ERP57, and also tapazin. And this tapazin molecule is really the molecular glue that helps to keep this complex together. It helps to bridge peptide receptive class 1 molecules onto the TAP transporter, bringing them close to the source of newly generated peptides. And tapazin is also a peptide editor, which means that it can help to load peptides onto the MHC class 1 molecules as well. So it keeps it in an open conformation, allowing it to be peptide receptive. And it was thought that after the MHC class 1 molecules had loaded peptide, that they went straight up to the plasma membrane for display and recognition by the immune system. However, when I was a postdoc in John Trousdale's lab at the University of Cambridge, we were interested in exploring whether there were any other components of the MHC class 1 antigen processing and presentation pathway that might have been missed. And we were particularly interested in TAPBPR. And the gene for TAPBPR was originally described to be encoded on the an MHC paralogous region on chromosome 12. And it was discovered in 2002, this gene, and it was shown to have 22% identity with tapazin. And it was predicted to fold into 3 immunoglobulin-like domains, like tapazin was too. And the TAPBPR molecule appeared to be polymorphic in this original description of it, with about 6 different alleles apparent. However, back in 2002, when the gene was first described, the function of TAPBPR was unclear. And in 2009, we set out to explore what the function of this TAPBPR protein was. And through our work over the years, we've discovered that TAPBPR combine to MHC class 1 molecules, and it is an additional component of the MHC class 1 antigen processing and presentation pathway. So given its sequence similarity to tapasin, we first asked whether TAPBPR could function as an MHC class 1 peptide editor or a peptide exchange catalyst. To explore this, we teamed up with Andy van Hattering and Tim Elliott when they were both at the University of Southampton to perform these peptide exchange assays for TAPBPR. So in these assays, we're asking, can TAPBPR help to dissociate peptide from MHC class 1 molecules, and can it help to promote peptide association onto MHC class 1 too? So when MHC class 1 molecules are loaded with a good fit peptide in these assays, they're relatively stable and the peptide is very slow to dissociate, shown here with this black line over time. However, we found that by adding TAPBPR into this system, that TAPBPR then caused the peptide on the MHC class 1 to rapidly dissociate, shown here with this, this blue line here. So we've got rapid dissociation of the peptide in the presence of TAPBPR in these in vitro assays. So this shows us TAPBPR can enhance peptide dissociation from MHC class 1. So what about this peptide loading step as well? So when you incubate MHC class 1, empty MHC class 1 molecules with peptides, they can bind peptides on their own, shown here with this black line. However, when we added TAPBPR into the assay, what we found was that peptides rapidly associated with the MHC class 1 molecules now. So much quicker peptide loading onto the class 1 molecules in these in vitro assays. So these assays told us together that TAPBPR was a peptide exchange catalyst that was capable of editing the peptides presented on the MHC class 1 molecules. So how does that translate into a cellular environment? When Clemens Herrmann was a PhD student in my lab, he knocked out TAPBPR in HeLa cells. And when we did this, we found that there was no obvious effect on MHC class 1 expression on the plasma membrane. So wild-type HeLa cells and our TAPBPR knockout HeLa cells express similar levels of MHC class 1. And we then teamed up with Stefan Stefanovic at the University of Tübingen to look at whether knocking out TAPBPR had altered the class 1 immunopeptidome. And this immunopeptidomic analysis revealed that the loss of TAPBPR caused a widening of the peptide repertoire displayed on the MHC class 1. So when TAPBPR is present, we have a restricted repertoire. When we knock it out, we expand the repertoire of the peptides. And this published data set, all of our published data sets kind of up to date, fit with this idea of TAPBPR presence causing the refinement in the immunopeptidome displayed on MHC class 1. So back in 2002, before we had AlphaFold, we worked with Janet Dean, who's a structural biologist at Cambridge, to produce a docking model of TAPBPR on MHC class 1. And when Janet did this, she identified a loop of TAPBPR, which she thought had the potential to interact with the MHC class 1 peptide binding groove. And this loop was comprised of residues 22 to 35 approximately of TAPBPR, and we predicted it was located near the MHC class 1 F pocket. And through Janet's work, she also suggested that tapasin contained a similar loop sequence, but in this case, the loop was significantly shorter than the TAPBPR loop. So we asked, does this TAPBPR loop play a role in peptide exchange? And to do this, Tudor Ilka, who was a PhD student in my lab, created a small panel of TAPBPR loop mutant proteins. So he first made what we call the dead loop variant, and in this variant we mutated all of the residues in that loop to a series of alanines, serines or glycine residues. And Tudor was also particularly interested in a leucine residue that was found at position 30 of this of TAPBPR in this loop region. So he made a specific mutant, L30G, in which he mutated the leucine at position 30 to a glycine. And then we also made a loop variant in which we had the dead loop, but then we reconstituted the leucine residue alone back into the TAPBPR loop region. And when we made recombinant versions of these proteins, differential scanning polarimetry revealed that all of the loop variants were stable and they had a similar melting temperature as the wild type TAPBPR protein. So we hadn't impacted stability of the TAPBPR. And then we also found that the TAPBPR variants with the loop mutations were still capable of interacting with MHC class 1. So when we immunoprecipitated TAPBPR from cells expressing these various TAPBPR variants, we found that the loop mutants could all bind to the MHC class 1 molecules. So we hadn't impacted its ability to bind to MHC class 1. We then went on to perform peptide exchange assays, looking at the ability of the loop mutants to perform peptide exchange. And this revealed that while the wild type protein, shown here in the dark blue colour, was very good at dissociating peptides from MHC class 1, when we mutated to the loop region of TAPBPR, we found we'd lost the ability of the TAPBPR to now promote peptide dissociation from the MHC class 1. And very interestingly, when we mutated that one residue in the loop, at leucine 30, so we mutated leucine 30 to glycine, again, TAPBPR lost its ability to perform peptide dissociation from the MHC class 1, suggesting that this leucine residue in particular was important in dissociating peptides. And then when we reconstituted the leucine residue back into the dead loop, again, we gained functionality of the TAPBPR dissociating peptides from class 1. Again, pinpointing that this leucine residue played a very important role in this process. We also went on to show that in cells, mutation of the TAPBPR loop region affects the immunopeptidome that's presented on the MHC class 1 molecules, showing that this is not just an in vitro assay system that this works in, but also in a cellular system. And this work led us to propose the following model for the loop being involved in peptide dissociation from class 1. So MHC class 1 molecules are known to breathe or relax in, and when they do this, the C-terminus of the peptide can partially dissociate. So we then think that the leucine residue of TAPBPR can be inserted into the MHC class 1F pocket, and that this then will inhibit the peptide from rebinding at the C-terminus. Then TAPBPR can cause further conformational change in the MHC class 1 that will prevent the peptide from rebinding to the MHC class 1 molecule, and then we have the complete dissociation of that peptide from the MHC class 1. Then in 2017, while we were doing this work on the TAPBPR loop mutants, Robert Tampe and David Margolesi's lab published back-to-back papers in Science revealing the structure of MHC class 1 with TAPBPR. And these new structural insights beautifully revealed that TAPBPR widens the MHC class 1 peptide binding groove. And this has led to new insights into how conformational changes induced by TAPBPR can cause low affinity peptides to dissociate from the class 1 molecules. Now, these structures revealed that the loop region of TAPBPR that we'd been working on was indeed potentially in close proximity to the MHC class 1F pocket. Although there's some different opinions to the precise sort of location of the loop on these class 1 molecules. So this loop region that we've worked on, thinking it's been involved in peptide loading onto MHC class 1 and dissociating the peptides, we like to think that the loop can fit inside the MHC class 1 peptide binding groove to function in dissociating peptides. But there's also the concept that the TAPBPR loop might function as a peptide trap that helps to push on peptides onto the class 1 molecules too. So there might be multiple different functions for this TAPBPR loop region in the editing process. Then subsequently in 2022, structural studies revealed a similar mechanism for tapesin-mediated peptide editing, with the tapsin loop region, and tapsin also containing this loop, a similar loop region, albeit shorter, which has a leucine residue, which has the potential to be interact with the MHC class 1F pocket too. So we now know that this TAPBPR molecule is an intracellular MHC class 1 peptide chaperone and a peptide editor that seems to be involved in a peptide selection step for MHC class 1 molecules. And we've also done work that's shown that TAPBPR can interact with an enzyme called UGT1 or UDP glucose glycoprotein glucosyltransferase 1. And when we get this complex, what happens is a UGT1 can change the glycan on the MHC class 1, which causes any empty MHC class 1 molecules to recycle back to bind to calreticulum, which then incorporates it back into the peptide loading complex for another attempt at gaining a peptide in this part of the cell too. So we now know that both tapazin and TAPBPR are capable of mediating peptide editing on class 1 molecules, and that they're working sort of mechanistically in quite a similar way. But that leads to a number of sort of outstanding questions. And one major question that we're interested in is why the MHC class 1 molecules need two different peptide editors that seem to be working in a similar way. And I don't think that's entirely clear yet. So could it be that these two editors are working in distinct sites? We know that tapazin works in the peptide loading complex, a nice peptide-rich environment, and that TAPBPR is playing a role not within this complex, at least in human cells. So, whether TAPBPR allows the class 1 to load peptides in additional cellular environments. Could it be that one of the editors, like tapazin, is predominantly involved in peptide loading onto class 1 molecules, and the other one, TAPBPR, would be involved in peptide selection or refining repertoire? Could it be that these two different editors exert different selection criteria on the MHC class 1, so maybe using their different loop regions to refine the repertoire in different ways? However, we were quite interested in whether different MHC class 1 molecules might use different editors. So could it be that some MHC class 1 have a preference for tapazin and other MHC class 1 molecules have a preference for TAPBPR? So to start looking at why these MHC class 1 molecules need two peptide editors, we first asked which MHC class 1 allotypes does TAPBPR bind to. Now, to do this, we made recombinant soluble TAPBPR and tested its ability to bind to a panel of 96 different classical class 1 molecules using the single antigen bead system that's commonly used by tissue typing labs. And this is a flow cytometry based assay that we used to determine if TAPBPR could bind to different HLA. And what this revealed was that TAPBPR had preference for binding to HLA-A molecules, particularly those of the HLA-A2 and the HLA-A24 superfamily, while we found it bound much less to HLA-B and C molecules. So we went on to test which MHC class 1 molecules our soluble TAPBPR was performing peptide exchange on, and found that the peptide binding strength roughly correlated with the ability of TAPBPR to promote peptide loading in these particular assays. So in these assays with recombinant TAPBPR, we could quite easily load peptides onto HLA-A molecules, but we found it much more difficult to load peptides onto HLA-B and C molecules. And then when we compared the sequences of the HLA-A molecules that our recombinant TAPBPR bound well to with those that the TAPBPR didn't bind well to, we found that the strong binders all had a histidine at position 114 and a tyrosine residue at position 116 in the MHC class 1F pocket. And when we reconstituted this HXY motif back into MHC class 1 molecules that TAPBPR bound weakly to, we found we could significantly increase the binding of TAPBPR by restoring this HXY motif. So here you can see TAPBPR binds weakly to HLA-A6801 molecules, B27 and B4405 molecules, But when we make a single amino acid mutation to ensure it has the HXY motif now in the F pocket, we now see much stronger binding of TAPBPR to these MHC class 1 molecules. And in contrast to that, when we take HLA-A6802, which is our MHC class 1 molecule that we've observed the strongest TAPBPR binding to, if we mutate the HXY motif by substituting the residue at position 116, we completely lost the ability of TAPBPR now to bind to that class 1 molecule. So this demonstrates that there is a significant influence of this MHC class 1F pocket in binding to TAPBPR. So we've shown that TAPBPR has preference for binding HLA-A2 molecules, particularly those of the A2 and A24 superfamily, and it binds relatively weakly to HLA-B and C molecules. And we can load peptides onto these HLA-A molecules, but we were really curious in what TAPBPR does, if anything, to the MHC class 1 molecules in which it binds weakly. So we turned our attention now to HLA-B molecules, which exhibited very low binding to the MHC class, to TAPBPR. And we decided to focus on HLA-B44 molecules, which exhibited extremely weak binding to TAPBPR. And we asked, does TAPBPR shape the immunopeptidome of these HLA-B molecules? And the reason for choosing B44 molecules is that they're very interesting because they lie at the opposite ends of the TAPSIN dependency spectrum. So B4402 is very dependent on TAPSIN for stable surface expression at the plasma membrane. However, B4405 lies at the opposite end of the spectrum and is relatively independent of TAPSIN for stable surface expression. And perhaps what's even more interesting is that these two MHC class 1 molecules, which lie at the opposite end of the TAPSIN dependency spectrum, vary at position 116, found in the MHC F pocket, which is where we found TAPBPR was also significantly influenced by. And again, this shows that minor variations in MHC class 1 can have a very significant effect on how they are going through the antigen processing presentation pathway. And what we were interested in addressing is whether there was an inverse relationship between TAPSIN-mediated and TAPBPR-mediated peptide editing. And we were curious whether these TAPSIN-independent alleles, such as B4405, were now reliant on TAPBPR for loading peptides, and whether the TAPSIN-independent alleles, such as B4402, were not edited by TAPBPR. However, there was nothing known about the effects of TAPBPR on the B44 immunopeptidome, so we decided to look at how TAPBPR shapes the immunopeptidome of B4402 and 05, which lie at the extreme ends of the TAPSIN dependency spectrum. Now, this work was performed by Orr, who was a PhD student in the lab, and the first thing she did was to confirm that in our cellular system, TAPBPR interacted very weakly with both B44 molecules in our cells. We then looked at the TAPSIN dependency spectrum of the B44 molecules in our HeLa cells, and Orr confirmed that B4402 was extremely dependent on TAPSIN for stable surface expression. Whereas B4405 was only slightly reduced when TAPSIN was absent. When we then depleted TAPBPR from cells, we found that this had no obvious effect on surface expression of either B4402 or B4405. So this suggested that neither of these B44 molecules were actually dependent on TAPBPR for stable surface expression. So then we performed an immunopeptidomics on our wild type and our TAPBPR knockout cells expressing the two B44 molecules. Now for B4405, what we noticed when we looked at the immunopeptidomes is that there was an enrichment in the tryptophan residues in the peptides that were enriched in wild type cells compared to the TAPBPR knockout cells. And when we did seq logo analysis, this revealed that this tryptophan residue was frequently found at the C terminus of peptides loaded onto B4405 in the presence of TAPBPR, but that this tryptophan residue was largely absent from peptides that were enhanced in the absence of TAPBPR. And this is the first time that we've observed a change in the sort of peptide binding motif specifically by TAPBPR. So we've previously found a sort of expansion of the peptide repertoire in the knockout cells, but nothing of particular interest when it came to the peptide binding motif. So this was the first observation that TAPBPR could have quite a profound effect on peptide binding motifs of an MHC class 1 molecule. And very interestingly, Melina Raghavan has evidence for tapism-mediated peptide editing on B4405 peptidomes. So what she found was, again, for B4405, that there was an enrichment of tryptophan residues at the C-terminus in the presence of TAPBPR. And Melina proposed that this occurred by tapism widening the peptide binding groove of this MHC class 1 molecule, allowing these bulky C-termini to be loaded. However, due to the extreme dependency of B4402 for tapism, Melina was unable to draw any conclusions about how tapism was shaping the B4402 immunopeptidome. However, luckily for us, B4402 is not dependent on TAPBPR for stable surface expression, so we were able to compare the peptide repertoires of cells with TAPBPR present and absence for these B4402 molecules. And what this revealed was that there was the tryptophan residue again was more common in peptides in the presence of TAPBPR on B4402, and then when TAPBPR was depleted, we didn't see this enrichment in the tryptophan residues at the C-termini. However, we didn't see quite as profound effects as for B4405, but this did suggest that, like B4405, TAPBPR was also promoting the presentation of a tryptophan residue on B4402. We went on to show that TAPBPR presence was improving the affinity of the peptides presented on both B4405 and B4402 molecules. And then, very interestingly, when we looked at the peptidomes, the breadth of the peptidomes, we made some interesting observations. So for B4405 expressing cells, what we found is that TAPBPR had an overall restrictive effect on the number of peptides that were presented. So in the TAPBPR knockout cells, we found this greater number of unique peptides displayed compared to the cells in which TAPBPR was expressed. And that fitted with what we found in the previous immunopeptidomic analysis. However, when we looked at the immunopeptidomes presented on B4402 molecules, we actually found something different that we hadn't observed before. We found that TAPBPR presence was having an overall expansive effect on the number of unique peptides that were displayed. So in the TAPBPR knockout cells, we found reduced peptide numbers compared to when the cells were expressing TAPBPR. And this was the first time we had observed this expansive effect for TAPBPR on any of the alleles that we had investigated to date. So finally, we wanted to validate the ability of TAPBPR to load peptides with various C-termini onto both B4402 and 05, using flow-based assays that we developed in which we overexpress TAPBPR in a cell, which brings up peptide receptive class 1 molecules to the plasma membrane, and then we can incubate these cells with fluorescent peptides and ask whether TAPBPR promotes their loading. And we looked at the ability of TAPBPR to load a model peptide, which had a glutamic acid at position 2, and then either a tryptophan, a phenylalanine, or a leucine residue at the C-terminus. So we varied the residue at the C-terminus of peptide and asked what effect that had on TAPBPR-mediated loading. And what we found is that for B4405, TAPBPR could load peptides onto these MHC class 1 molecules. And we found that TAPBPR appeared to load peptides with a C-terminal tryptophan or a C-terminal leucine more readily than it did with a C-terminal phenylalanine. But rather surprisingly, despite seeing that expansive effect for TAPBPR on the B4402 immunopeptidome, we found that TAPBPR was not efficient at loading peptides onto this MHC class 1 molecule. So we could only see a little bit of peptide loading in the system at very high concentrations of peptides for peptides that contain the C-terminal tryptophan. So how does this data on these B44 molecules fit together with the known literature? Well, B4405 is known for its ability to bind peptides in a tapesin-independent manner. And this will result in a population of B4405 molecules which could be loaded with suboptimal peptides. And we propose that TAPBPR is playing an important role in mediating the removal of these suboptimal peptides from B4405 molecules due to its action as a peptide exchange catalyst. And this fits with the overall restriction in the peptide repertoire we observed in the presence of TAPBPR, and also the ability of TAPBPR to perform peptide exchange on these MHC class 1 molecules. So we think the editing role of TAPBPR is really important for B4405. In contrast, B4402 is known for being very highly dependent on tapesin for mediating peptide loading. And our findings really suggest that TAPBPR is not particularly good at loading peptides onto B4402 molecules, although it somehow helped to broaden the peptide repertoire displayed. So how does that make sense? It's not able to load peptides, but it's broadening the repertoire. And what we propose is that for B4402, TAPBPR is predominantly functioning as a chaperone, stabilizing B4402 in a peptide receptive state. And this subsequently helps to push the B4402 molecules onto tapesin for loading in this nice peptide-rich environment of the PLC. So different functions of TAPBPR for class 1 molecules with different variations in these B44 molecules. And recently, we've also expanded our work into non-human TAPBPR by exploring the function of mouse TAPBPR. And this revealed that, like its human counterpart, mouse TAPBPR could load peptide onto mouse MHC class 1 molecules in our assays. And very interestingly, when we looked at our immunopeptidomic analysis, comparing the peptide repertoire displayed in wild-type cells with TAPBPR knockout cells, what we again see is that TAPBPR presence was having an effect on the C-terminus of the peptide. And for H2KB molecules in mice, we found that murine TAPBPR presence was promoting the presentation of peptides with a C-terminal methionine residue. And we also went on to show that mouse TAPBPR could load peptides with the C-terminal methionine residue as efficiently as it could load peptides with a C-terminal leucine residue. So it's not just the B44 molecules that TAPBPR is having an influence at the C-terminus. We're seeing this now potentially more commonly across TAPBPR. So we've still got to do lots of other class 1 molecules, but you can see here that now we are seeing a trend in TAPBPR shaping the C-terminus of the peptide. So just to summarize this part of the talk, like tapesin, TAPBPR is an MHC class 1 intracellular peptide editor. It exhibits prolonged binding to HLA-A molecules, and this seems to be determined by residues in the F pocket. It binds weakly or transiently to most HLA-B and C molecules. However, despite this, it is able to shape the immunopeptidome of the MHC molecules that it binds weakly or transiently to. And we're now seeing an effect of TAPBPR on the peptide binding motif of MHC class 1, in particular on the C-terminus of the peptide. And this TAPBPR editing loop region seems to be playing an important role in peptide selection on MHC class 1. So now I want to move away from the MHC class 1 polymorphisms and turn to the TAPBPR polymorphisms. So it turns out TAPBPR is also polymorphic, so we wanted to explore how these polymorphisms might affect its function. So analysis of the 1000 Genomes Project revealed that there are 6 common TAPBPR alleles that cover approximately 97% of the haplotypes in human populations. And for us, it turned out that allele 29 was the wild-type TAPBPR molecule that we've been working with in the lab. So we were working with a very common TAPBPR variant that we've used in all our assays to date. And when we looked at these 6 common TAPBPR alleles, we could see that the variations that we find in them are relatively subtle, and that they're found right across the whole of the TAPBPR molecule. We've then went on to explore the functionality of these common TAPBPR molecules, and we found that they all bind to MHC class 1 to a similar extent. So this was using assays in which we overexpress the different variants, and then immunoprecipitate TAPBPR from the cells, and then do a Western blot for MHC class 1. So we didn't see any particular change in the ability of these common variants to bind to MHC class 1. Then we also performed peptide exchange assays on a couple of different class 1 molecules, and we found that these different common variants of TAPBPR could all mediate peptide loading onto class 1 molecules to a similar extent. So this work suggests that the majority of individuals have a TAPBPR molecule that have similar functionality. So then we decided to explore some of the function of some rarer TAPBPR variants that we sort of cherry-picked because we thought they would look particularly interesting. So for example, we looked at allele 32, which is the histidine to tyrosine change at position 26, which is within this TAPBPR peptide editing loop region that we recently talked about. We were also interested in allele 27 and allele 7, which looked like they had the potential to be glycosylated due to the variations introducing an NXT or an NXS motif into the TAPBPR molecules. And we also picked another couple of variants that we thought looked interesting due to specific changes. Now, first we made recombinant soluble variants of these rarer TAPBPR variants. And what we noticed is that the 293T TAPBPR variant was running at a higher molecular weight, suggesting that it was glycosylated as we'd predicted. And in the treatment of this protein with an enzyme called PNGSF, which cleaves N-like glycans, confirmed that this added molecular weight was indeed due to the 293T variant being glycosylated. So we see this reduction in its size upon treatment with the enzyme. We then explored the binding of the different recombinant TAPBPR molecules onto the panel of the MHC class 1 molecules using the single antigen bead system again, comparing the wild-type TAPBPR molecule shown here in white with the variants shown in different colours. And very interestingly, we found that the 293T variant shown here in orange had a significantly reduced ability to bind to MHC class 1 molecules on these single antigen beads. So we did observe a little bit of binding to HLA-A6802 and HLA-A2, but other than that, it was relatively flat for binding to MHC class 1. In contrast, the H26Y mutant in that loop region seemed to show increased binding to a variety of different MHC class 1 molecules, shown here in the blue bar. So this suggested that this H26Y was exhibiting increased binding compared to the wild-type TAPBPR. And we went on to confirm these results using cell-based assays, and this confirmed that the A293T variant had a very reduced ability to bind to MHC class 1 molecules, whereas the H26Y variant exhibited increased binding to MHC class 1 compared to wild-type TAPBPR. So to explore why this 293T variant exhibited such reduced binding to MHC class 1, we turned to AlphaFold. And here you can see the structure of the wild-type TAPBPR bound to MHC class 1 that's generated via the AlphaFold. And then we used AlphaFold to model the position of the N-link glycan on TAPBPR on the 293T variant. And what they showed is that 3 of the 5 models that were generated via AlphaFold predicted that the glycan would point out in the direction of the MHC class 1 molecule. Now it's worth noting that AlphaFold currently can only add glycans with 8 sugars attached, and the one on TAPBPR, which would be present in the ER, we would predict to have to be bigger and would likely be comprised of 11 or 12 different sugars, so it should be substantially bigger than what we've modelled here. And then when we overlaid these 2 structures, that's this wild-type TAPBPR with class 1 with our 293T variants, it's clear that the glycan clashes with beta-2 microglobulin. And we think that this explains the reduced binding to MHC class 1, that the glycan is sort of sterically hindering the ability of TAPBPR to bind to MHC class 1. And here, if we zoom in, you can see that this glycan, these glycans here, so this is 3 different models of TAPBPR with the glycan, really do impact its ability to bind to beta-2 microglobulin. And there's lots of different clashes that are observed here with the glycan with specific residues on beta-2 microglobulin. So the glycan that's introduced on the A293T variant is predicted to clash with residues at position 42, 74, 75, 76, and 77 of the beta-2 microglobulin domain on the MHC class 1. So now turning back to the H26Y TAPBPR variant, we used SPR to look at the affinity of this variant for MHC class 1, and we found that it had higher affinity for class 1 compared to wild-type TAPBPR. So to explain this, why is this H26Y variant got higher affinity for class 1, we've turned to molecular dynamic simulation just recently to explore this. And what this suggests is that the introduction of this tyrosine residue stabilizes the TAPBPR editing loop. And it now forms, this tyrosine residue forms stable interactions with alanine 29 of the TAPBPR loop, and also a tryptophan residue, which is found at position 147 in the MHC class 1 alpha 2 1 helix. So this thereby bridges this loop region closer to the MHC class 1 F pocket. In contrast, we've found with wild-type TAPBPR, the histidine residue at position 26 in the wild-type molecule makes it more flexible, and the histidine is predicted to make transient interactions with lots of other residues in the TAPBPR editing loop. So we see lots of different interactions happening here, and we think that this actually positions the loop region higher up in the peptide binding groove than with the H26Y variant, which seems to keep the loop quite low and bound to the MHC class 1. So following on this, we wanted to understand how these 2 natural variants fare in the peptide exchange assays. So to test this, we turned back to our recombinant TAPBPR assay system and asked, how does the 2 variants perform in these exchange assays? And what we found is that when we use this recombinant soluble TAPBPR, the A293T variant with the glycan and its reduced binding to class 1 didn't perform peptide exchange very well. So it was very, very weak at performing peptide loading onto MHC class 1 molecules. In contrast, our H26Y variant with its increased binding to MHC class 1 seemed to be better at performing peptide exchange, and we were observing increased binding with the recombinant TAPBPR system. However, when we then use an alternative assay in which we overexpress full-length TAPBPR in a cell, which brings this peptide receptive class 1 up to the plasma membrane, we then found different results with our variants. In this case, the 293T variant, which has weaker binding to MHC class 1 was actually better at loading peptide onto the class 1 molecule. So we think this is because it has lower affinity. It can then be more easily outcompeted when we add exogenous peptide. In contrast, the H26Y variant with the higher affinity for MHC class 1 found it more difficult to outcompete with peptide, and this resulted in its lower ability to load peptides onto these MHC class 1 molecules in this assay system. So regardless of these different results for these different assay systems, these results show that there are rare hyper and hypofunctional variants present in the human population, which has different binding affinity for MHC class 1, which then results in different functional consequences for peptide exchange. So in the last few minutes, I just want you to explore some data that we had that shows, asks, is there a therapeutic potential for TAPBPR in cancer? So going back to these assays, when we used recombinant TAPBPR, we found we could use it to promote the loading of tumour-derived antigen and viral peptides onto tumour cells in vitro. So here you can see examples where we loaded HLA-2 molecules that are expressed on the breast cancer cell line MCF7, and we could load peptides derived from P53 and HER2, and we could also load peptides derived from human cytomegalovirus and also Epstein-Barr virus. So this got us wondering whether recombinant TAPBPR could be used to make tumours more immunogenic. And we're particularly interested in so-called cold tumours, which have low immunogenicity and therefore not recognised by CD8 positive T cells. And this makes these tumours unresponsive to immunotherapy. And actually, in recent years, we now appreciate there's actually a significant number of viral-specific T cells that are found in tumours. For example, EBV, CMV, and flu-specific T cells have been found in a wide range of tumours, including lung, colorectal, and melanoma. And these viral-specific T cells in tumours are described as bystander T cells that lack the signs of classic exhaustion. Therefore, given our ability to use recombinant TAPBPR to load peptides, virally derived peptides onto tumour cells, we wanted to explore whether there was a way to target TAPBPR specifically to tumours. And then we could use this to load exogenous peptides and to activate these viral-specific T cells within the tumour microenvironment. So Andreas Nerinx, who was a postdoc in the lab a number of years ago, made a series of TAPBPR fusion proteins in which we fused the luminal domain of TAPBPR with either a nanobody or a single-chain variable fragment to try to tether the TAPBPR to a target protein of choice that we would express on the surface of tumour cells. And we found that using this system of tagging with an antibody, we could deliver TAPBPR fusion proteins to the plasma membrane of cells expressing the correct protein for the antibody fragment. So here, for example, you can see TAPBPR delivery using a HER2 single-chain variable fragment to cells expressing HER2. So having achieved this targeted binding of TAPBPR to the plasma membrane of the target cells, we then asked whether the TAPBPR domain was functional and was capable of promoting peptide loading onto MHC class 1. And we found that this was indeed the case, that our TAPBPR fusion proteins were extremely efficient at loading peptides onto tumour cells expressing the target protein, but it didn't load peptides onto cells that lacked the nanobody or the single-chain variable fragment protein. And here in this line graph, you can see just how efficient these TAPBPR fusion proteins are at loading peptides onto target-positive tumour cells. So we can load peptides when we pre-treat cells with TAPBPR fusion proteins for just 15 minutes, followed by incubation with peptide for 15 minutes. We actually see an incredible amount of peptide loading onto the peptide tumours. And this means we can really dial down the amount of peptide needed to decorate tumours with peptides. So really we can go down to the picomolar levels and hopefully still obtain, manage to achieve decoration of tumours with peptides. We've also tested whether it's possible to deliver TAPBPR fusion proteins onto tumours in vivo. Now, to do this, we had mice that were bearing subcutaneous tumours expressing their target protein, and then we IV infused them with our TAPBPR fusion proteins. We then harvested the tumours and the organs from the animals between 15 minutes and 6 hours post-IV infusion. And then we would probe for TAPBPR presence. Our data suggested that TAPBPR was specifically targeted to the tumours in vivo. So it was detected in the tumour just 15 minutes post-IV infusion, and it was still present in the tumour 6 hours post-IV delivery. In contrast, we were unable to detect our human TAPBPR in highly vascularized organs such as the lung, and our data suggested that TAPBPR fusion proteins that were in excess was being cleared by liver metabolism and renal clearance. So this data suggests that we can target TAPBPR to tumours, to the plasma membrane of tumours in vivo. We next tested whether the TAPBPR that had been delivered was indeed functional by performing peptide exchange assays. And what this revealed is that we could load exogenous peptides onto tumour cells that expressed the target, but that the cells that lacked the antibody target within the tumours were not decorated by peptides. So this really suggests that these TAPBPR fusion proteins are functional when they're delivered to tumours in vivo. If these fusion proteins are acting as we hope they are, the next functional consequence should be the activation of viral-specific T cells by the tumours. And that's what Orr saw when she went on to perform some T cell assays using CMV-specific T cells in collaboration with Mark Wills in the Department of Medicine at Cambridge. So here you can see some data looking at interferon gamma secretion, for example. So when we treat our tumour cells with picomolar concentrations of peptide alone, it's not enough to trigger the T cells to respond. However, when we pre-incubate the tumour cells with our TAPBPR and then incubate with the peptides, we now see that the T cells are activated and that they secrete interferon gamma. And actually the level of the T cell activation we can observe is similar to when we express the viral protein inside the tumour cells itself in their natural percentage, shown here with the yellow bar here. So this is really demonstrating that we can use these TAPBPR fusion proteins to load peptides onto the surface of cells, and the T cells can then respond to them. Now, Orr also demonstrated that we can induce these viral-specific T cells to degranulate when we incubate the target cells treated with TAPBPR fusion and the viral peptides. So we actually have a functional consequence of T cell degranulation. And we've also gone on to show that the TAPBPR fusion treated cells, the tumour cells, are specifically killed by the T cells too. So taken together, our work suggests that these TAPBPR fusion proteins could be used as a way to override the natural antigen presentation pathway and be a way to turn cold tumours into hot tumours by decorating them with peptides. So on healthy cells, there's no antibody target, and as a result, this antibody fusion protein doesn't bind. And we actually found that the antibody fragment binds to the MHC, the TAPBPR, and masks the MHC class 1 binding site, which is why it doesn't bind to target negative cells. However, on cancer cells, which express the target protein that the antibody domain recognizes, this then causes the TAPBPR fusion protein to bind via the antibody fragment. This then releases the TAPBPR fragment. It can bind to MHC class 1 on the plasma membrane of tumours and cause peptide dissociation. And this leaves the class 1 in a peptide receptive state. And then when we incubate with our fluorescent peptides or peptides that the T cells can see, we can then get loading of the antigenic peptides onto the MHC class 1, which subsequently means that the tumour cells can be recognised by cytotoxic T cells causing cancer cell death. So that just leaves me to thank all the people who have been involved in this work. The work on TAPBPR was started by John Trousdale when I was a postdoc in his lab. We've been helped by a number of collaborators at Cambridge, both structurally and using T-cell assays and in vivo work. We did TAPBPR peptide exchange assays initially with Tim Elliott and Andy Van Hattering when they were both at the University of Southampton. They're now both in Oxford. We've been helped by a number of people at the University of Tübingen over the years, first by Stefan and now by Jens and Julianne. TAPBPR specific antibody reagents have been made by Karsten and Mikael. And then I've highlighted that a lot of the people in the lab who have been specifically involved in this work, but we've over the years had a number of people working on TAPBPR, master students, PhD students and postdocs who've done a brilliant job at characterising TAPBPR. And finally, I'd like to thank our funders, the Wellcome Trust predominantly, and we've also had some money from CRUK and also the MRC. And thank you for your attention today, and I'm very happy to take questions.
Oh, thank you, Louise. That was wonderful. We have a few questions in the Q&A, and I gave Gurvinder and Martin the ability to talk so they can just ask their questions instead of not having.
Lovely. Do you want me to stop sharing my slides? Yeah. So.
Yeah.
That good? Yeah, we're good. Yeah.
Okay. Well, this is Martin Myers.
Hi.
Thanks for your talk. That was wonderful. So I'm wondering about this impact of these TAPBPR variants on the T cell repertoire, and whether two individuals with the same HLA genotype, but only the difference in this, in these variants, would that possibly be observable in the repertoire, and could that be something that has some clinical implication, for example, graft versus host disease or some other transplant?
Yeah.
Kind of
We've
target?
not. What we're trying to do is look at now whether some of the TAPBPR variants are associated with inheritance of a particular MHC. So there's a lot of work, I think, to do to understand these variants and whether they're associated with particular MHC, whether they can promote peptide loading better on different MHC molecules. We're really just scratching the surface. There's so much that we could do. We're starting to look at whether there's disease associations as well. So we're starting to do that work with people in Oxford, looking to see whether, is it disease association with any of these variants. So yeah, lots of questions there. We're really just beginning to scratch the surface at what these variants might be doing. We've not even done the immunopeptidome yet of these different variants, which I think that'd be something that's very interesting that we should look at first.
Yeah, they more directly, than the TCR.
Yeah.
Thanks.
Thanks.
Was there a different question, Steve?
Yeah, we have a question from Malcolm Sim and also Urban Power still. So if either one of you wants to pipe up. Okay, well, Malcolm says, what's the plan to direct the peptides to the tumor in vivo? Oh, Malcolm's on a bus, so can't talk.
So can you repeat the question?
What's the plan to direct the peptides to the tumor in vivo?
Yeah, there's not a so we have done some work where we've done intratumoral injection. I think that is the difficult part of the system. It's not that we can deliver the TAPBPR to the tumor, but ensuring there's enough peptide there to load onto the tumor is the difficult part with the short half-life of the peptides. So we have done some intratumoral injection in vivo and found we can load peptides onto the tumor cells, but obviously that's not applicable to all tumors. So the idea would be, can we deliver peptides IV and would they get there? But that's the difficult part, I think, of the system, is whether we can actually, if the peptides would be stable long enough, whether you can engineer the peptides so they have increased stability in vivo. So yeah, that's a challenge.
Okay, and let's see. Gurvinder Cow asks, what is TAPBPR analogy in pig?
In pig?
I guess analog, yeah.
We've only just looked at mouse, so we've just done human and mouse so far. I know that Peter Cresswell is quite interested in looking at TAPBPR in bats, so there is some work going on looking at TAPBPR in bats, but I don't know anyone else doing any other work on TAPBPR in different species. So yeah, there's been some work on chicken too. So there's chicken TAPBPR. Next Garakis has looked at chicken TAPBPR and shown that has quite high affinity for MHC class 1. So it's higher affinity than the human version. So chicken's been done, mouse has been looked at, and yeah, human. Lots more to do.
Let's see. They also asked, why was the L30G position selected to start with?
Yeah, so it's actually, so Tudor Ilka had been in Sebastian Springer's lab and as a master's student, and they were working on dipeptides with like a GL that they found bound to the F pocket. And then when Tudor came across to do his PhD in Cambridge and we were working on this loop, he said that's a natural dipeptide that's in that loop region. So he was very focused on the leucine residue. I didn't think it was interesting at all. You know, I thought it's all these charge changes that are going to be interesting. But he, because his work, the previous work on the dipeptides, was interested in this leucine residue, and he said it should bind to the MHC class 1F pocket. So that's why we went after the leucine residue specifically. Yeah, it came from Tudor, not me.
And I have a question. Do you have some idea why TAPBPR is in not in the MHC proper? Do the low variants that you find, do they have some sort of advantage maybe in some context?
Yeah, I don't know. I mean, I guess we assume that because TAP is in, we've done lots of work on that, that's the first editor, you know, and that's the main editor. I guess if you look at evolution, you know, could TAPBPR have been the first editor and then TAP is in evolved from that? I really don't know. I'm not an evolutionary biologist. But, you know, it's interesting to think that whether TAPBPR was the prototype and then TAPism arose from that. Yeah, I don't know why it's in a different MHC. I mean, ERAP, they're in a different region. Beta-2 microglobulins on a different chromosome. So there is a precedence for key components of the pathway not being within the MHC. But what that means in terms of advantage, yeah, we don't know yet.
Right. Well, again, thank you very much. Where'd my notes go? Hold on a second. I just want to remind everyone that you presented this work at our second annual symposium in Sassenheim last fall, and we're going to be having our third annual symposium this year on November 2nd to 4th, and everyone can register by visiting sip-sassenheim.org or immunepolymorphismsociety.org. So thank you for a wonderful presentation. Thank you all for attending. And we're all going to have more webinars over the rest of the fall.
Thank you very much.
Thanks. Bye, everyone.
Bye.